Okay, well, it's my great pleasure to introduce this year's recipient of the SDB Elizabeth D. Hay New Investigator Award. This award was named for Dr. Betty Hay. She was a pioneer in cell and developmental biology. She studied limb regeneration, epithelial to mesenchymal transition, and the role of extracellular matrix on cell differentiation, topics which I heard people give talks on multiple times during this meeting. She served as the president of the SDB. She was the editor for the journal of our journal, Developmental Biology. What? Whatever Ida tells me, I do. And she was a member of the National Academy of Sciences. I got it out of Wikipedia. It must be true. This award recognizes new investigators who have performed outstanding research in developmental biology during the early stages of their independent career. And so this year's recipient is Dr. Mubarak Syed. He's from the Department of Biology at the University of New Mexico in Albuquerque. Dr. Syed studies mechanisms regulating neural diversity and function. He uses Drosophila and investigates neural glial fate specification and interlineage communication. Dr. Syed is originally from Kashmir, India. His origin story to enter the field of science is one of curiosity, passion, persistence, and hard work. I hope he shares some of his origin story with you. Beyond his research, he has been very active in science outreach. He created something called JK Scientists for Kashmiri students. And in New Mexico, he created Pueblo Brain Science, providing hands-on neuroscience experiences for Native Americans and rural students. He's also an active contributor to the SDB. He was one of the co-organizers of the SDB Southwest Regional Meeting that was just last month. Actually, reading all the stuff he does, I got tired because he's so active and creative. Dr. Syed, please come up and give your talk.
Okay, thank you. Thank you very much. Thank you, SDB, for this great honor. And I cannot believe that I have been chosen for this award. There are so many extraordinary scientists, and thank you very much. And today's talk, I will talk about developmental story of my journey into science, what I do as a scientist, and how I train the next generation of scientists, mostly developmental biology with a small, little flavor of neuroscience. So before I begin, I want to acknowledge the native people who have lived here in Samoa. And thank you to all my mentors, teachers, collaborators, friends, and family. I have been reading about Professor Elizabeth Hay. It's an extraordinary honor. I wanted to see how I relate to her. She has been extraordinary. I think there are some similarities which I will connect or try to connect from my talk. One is love of microscopy, which is for her electron microscopy, for me mostly confocal and with small flavor of electron microscopy as well, and education and mentoring. So how many of you know Kashmir? I mean, this is disputed, has always been disputed, I don't know, many, good proportion. So this is where it is. We call it Indian-controlled Kashmir. So there's also other part. It's a small, beautiful valley in the mountains, very beautiful. This is our village, but always kind of in news. So where did it all start? It all started with my father and mother. On my right side is father, who unfortunately passed away in 2008, and on the left side is my mother. Although both of them had no formal education, but very open-minded, very down-to-earth people. And then As you know, Kashmir has always been in trouble, so my childhood, my youth, all was during war. And after education, mostly in university, mostly at Holmes College, I decided to move out. That's where I began my journey. This is how Kashmir will look like, mostly beautiful mountains. And my entry into developmental biology is through this gentleman who unfortunately also passed away on the right. How many of you recognize that person on the right? Professor Benishilo, he's at Weizmann. He used to come to India every 2 years, organize developmental biology workshop. So I was at NCBS and I applied to the course and I got into the workshop. 15 days, free food. It was amazing, right? Then I got to see flies, I got to see zebrafish, and then during that time, I met again extraordinary developmental biologist who also unfortunately passed away a few years ago, Veronica Rodrigues. You know Veronica Rodrigues? I mean, an amazing developmental biologist. And on the right side, here left side, is Professor Vijay Raghavan, again a developmental biologist. So I got into developmental biology through the mentorship of amazing people and scientists. This was the lab picture then. Then I moved to West, from East to West, and this is, again, extraordinary developmental biologist here, Christian Klemp, on the top here. I cannot see, the cursor is not working, but you can see here on the right side is Christian Klemp. And I did my PhD there, 4 years. And then I met again, I think these workshops are very important. I met Chris Doe during Okinawa workshop, developmental neurobiology workshop. Professor Noza is here. He was also one of the organizers. Then I decided to do my postdoc with Chris Doe at University of Oregon, who also got Conklin Medal last year. Amazing mentor. And this is, again, my mentors. How many of you recognize the other guy? Claude, yeah. Claude Diplan I met during postdoc. We stayed connected. He has been an amazing mentor also throughout my postdoc and early career in my life as a faculty. He also got Conklin Medal. So amazing mentors who have to shape the next generation developmental biologists. So during my 6 years at UNM, I have been lucky to, again, meet amazing scientists who are mostly neuroscientists. They study behavior, and they also have been amazing mentors. So Kathy, Matt, Vivek, and Monica, so they are all collaborators. So kind of, when I was in Chris's lab at the end, most of the developmental neuroscience in flies turned to circuits and behavior. I was kind of interested in linking development with behavior, and I had to reach out, and I got lucky to get these amazing collaborators. So that's what we study, development of behaviors. So this is our lab. And it snows in New Mexico. Lots of undergrads, so it'll be mostly undergrads and PhDs.
What? It's Friday, man.
So today I'll be talking about their work. So I'll talk about 2 projects. Well, thank you. This is a movie shot by talented undergrad Krishna. So most of the work we do is with undergrads, and I have two graduate students who graduated, which I will present today their work. So what we are interested in understanding how brain generates this amazing diversity. There are billions of neurons, more than 80 billion, of thousands of different kinds, and these cell types self-assemble to form circuits which drive complex behaviors. It all starts during development when these stem cells, here for example, outer radial glia, they divide over time, and based on the birth order, they can end up in deeper layers or superficial layers. The ones which are born early stay closer, end up in deeper layers, and the ones which are born later go to the superficial layers. So kind of time dictates what kind of neuron or cell types are being formed over time. So this is very important. We all, as a developmental biologist, know if something goes wrong during this critical period, during neurogenesis, we end up with severe neuropsychiatric and neurodevelopmental disorders, and many of them come up with these behavioral deficits, including sleep disruption. So what model system we study? We study insects, and insects, this amazing capability of navigation. This happens through a brain region called central complex. Central complex is higher order brain structure of insects. It's conserved from grasshoppers to fruit flies. Although the structure may be different, but the function and the neural structures are conserved. It's very important for sensory motor integration, for navigation, for internal state, many complex behaviors, and also sleep. So looking at the fruit fly, although small, tiny, has complex brain, does complex behaviors. Here is a central complex, which has 4 neuropil structures. The donut shape, which is kind of a donut, is called ellipsoid body, very important for spatial navigation. Then there is mustache shape called protocerebral bridge. And then there is fan-shaped body called fan-shaped body, and there is pair of noduli. And these 4 structures altogether, our neuropil structures, form central complex. So now we have amazing technology. That was my kind of tribute to Betty, having connectome picture here. So this is the connectome of fruit fly, which is freely available, one of the complex organisms where we have fully annotated connectome. So if you type central complex, this is the fly wire, you get all these neurons which have been associated with central complex. There are about 3,000 neurons of 255 different subtypes. And broadly, you can categorize them into columnar neurons, which divide the central complex into columns, and also tangential neurons, which have cell body outside and project to the fan-shaped body and make distinct layers of the fan-shaped body. So in a way, our cortex is divided into columns and layers. Similarly, the central complex is divided into columns and layers. And there are also these circular neurons called ring neurons, which you can see here at the bottom. Okay, so what do? We study maggots, because maggots are the ones where stem cells divide and make the adult fly brain. And this is now a schematic of this very important slide. There are 2 major subtypes of stem cells, neural stem cells. One is called type 1, which is red here. Type 1 self-renew and generate differentiated progeny over time. If they divide during maggot life, we'll make 100 neurons. But we care more about these blue ones, which are called type 2. They divide in a unique way because they not only self-renew themselves, but also make intermediate neural progenitors, INPs, which also act as stem cells. In a way, they diversify and make more neurons and glial types, although only 8 in number. So there is a similarity of these type 2s to human outer radial glia, which also make INPs and generate central complex. And this picture here is taken by permission now of my first kid. It's a fetal brain. And he grows up to here, attests that fly is a good model system to study development. He is here, but maybe not here. They are both here today. Anyway, so overall, our goal in the lab is to understand how during larval development, these stem cells will divide, what are the molecular programs which generate these distinct classes of neurons of the central complex. So this is my 6-year postdoc work with Chris Do in one slide. Early on, Chris Do has shown like these Drosophila embryonic stem cells express different genes over time called temporal identity. Later on, similar programs were shown to be important in the vertebrate brain development. But when I joined the lab, I did larval. I studied larval type 2 stem cells and showed that there are early expressed genes, late expressed genes. What was important is that late stem cells express receptor for hormone. As a result, insect hormone also acts on these stem cells to define temporal patterning and also perhaps in the future, which I'm doing now, different cell types and behaviors. So overall, if we focus today on one single stem cell called DL1, which we'll talk about a little later also, DL1 is the one on the side. How does DL1 over time define these distinct tangential neurons which end up in the central complex. And specifically today, we focus on fan-shaped body. Also, do you know this guy? Many of you have seen this guy. The top one. So this is Chris Doe from left to right. The days when life was fun, funding was available, life was good system. And today is here. So this is kind of a lineage of Chris Doe. And I don't know how many are here. 1, 2. Michael is here. Michael Hayden. Anyways, he has been extraordinary mentor, right? So we got what we got from him. So this is kind of take-home message. When we talk about self-hate, when we are born, where we are, what we associate with, we get lots of things from them. And he has, I counted last time, more than 40 faculty and many more other scientists in academia, industry as well. Amazing mentor. Again, kudos to Chris Doe. Okay, so DL1 is one of the 8 you see on the top right. That is called DL1. And now with Connectome, again, power of developmental biology, we can assign clones even in Connectome. This is similar to what we generate in the lab. So each DL1 makes about 136 neurons. And there are also glia, which are a separate topic. So today's work, we will talk about one subclass of these DFBs in 138, which are called here sleep-promoting neurons, the ones in green. They will inhibit helicon cells, and the result, the fly will sleep. And the work is by Adil, who graduated recently on the left, and Gonzalo, who is now a PhD with Chris Doe. So I have made a pipeline sending back to Oregon. And the other undergrad also, Alexa, is also with Mark Freeman in Oregon. So this is sleep-wake circuit. The green ones are DFB neurons, which we will talk a lot. And on the right side is connectome. Okay, the first thing we wanted to know is if these neurons come from type 2 stem cells. So this is intersectional genetics. If you want to know the answer, we can talk about later if you want to know more details of how it works. But if there is color, like here in green, that means they come from type 2s, right? Any of these 16. That was interesting. So then the genetics we have, thanks to Shumin Lee, who has made amazing tools from Markham to now CLIN, we can use CLIN to assign what stem cell makes these neurons. You see here red, and in the optic lobe glia, that means this is a DL1 clone. If you see green, that means these green neurons come from this stem cell. So we can track back lineages from adult to larvae. So majority of these neurons, which are sleep-promoting neurons, come from DL1. And what was interesting is although they look similar, this neuron also green here, but it's different because it comes from a different stem cell, which is called DM1, which is in the middle top. So although they may look similar, but if we do more profiling, maybe more molecular markers, they may be very different from each other. Again, we wanted to know in the adult, at the bottom here, these neurons express a gene, which is in science called E93, aggressor-induced gene. And I had found in my postdoc that E93 on top right is expressed in stem cells, late stem cells. So we thought maybe late stem cells make, these DFB sleep-promoting neurons. That's what Adil did. So we did birth dating. This is genetic birth dating. You heat shock over time. And if you see color, you see if you heat shock at 0 hour, if they are born later, you see color. If you heat shock 48, if they are born later, you see color. If they are born early, you don't see color. That's how you get the genetic birth dating. So you generate clones. You can see here when we heat shock 0, when adult heat shock 0 hour, 48 hour, you see them, but not after 76 hours. That means they are born during larval development from those two stem cells between 48 and 76 hours. So that's again a quote. Tell me whom you associate with, and I will tell you who you are. So as I mentioned in my postdoc, there are early factors, there are late factors, and these neurons seem to be born late, so we thought maybe late factors are important for the specification of these neurons. That's what we tested. So we are doing manipulations in maggot brain, in larvae, and we are seeing adults here. And the green is again sleep neuron, and magenta is NC82, which is general brain morphology, to see general brain morphology. So this is the control brain, and this is ECR knockout. So if we do ECR mutant in those stem cells, these neurons are not specified. That means ECR is necessary for the formation of these neurons. So again, if you read the developmental biology Gilbert textbook, 3 experiments you do. Find it, lose it, move it. So this is find it, and then first was find it, then this is lose it. So you lose it, you lose the gene function, you lose the neurons. So there was some cases where some neurons survived, but they were mistargeting to different layers. This is not on the slide. So kind of here early on, these stem cells on the model, they don't express ECR. Later on, they express ECR. Then ECR, through hormone excitation signaling, is important for the formation of these neuron types. ECR, again, is a signaling molecule. It activates many genes and represses many genes. We wanted to know which among these is important for the formation of these neurons. And we focused on E93 because E93 was present in the adult sleep-promoting neurons. So our hypothesis is this excitation regulates DAP fate via E93. So again, we are doing manipulations in larvae and studying here, assaying adults. So in control, you see normal morphology and normal numbers. When you do E93 loss of function, or here knockdown with RNAi, you see these neurons are missing, are not specific. So it's necessary, E93, but not sufficient, because when you misexpress, you don't get more of them. That means something else is also important for the formation of these neurons. Okay, so these were all 8 and 816 stem cells, but with fly genetics, we can narrow it down to one single stem cell. That's what we did here. 17A12Gal4 is expressed in DL1 only. So if we do DL1 manipulation with either E93 or ECR, sorry, this is ECR flipstop, we have the same phenotype. That means excitation signaling in those stem cells is important. So it is cell autonomous in DL1. It's very important in that stem cell. And there are some neurons which survive here, but since we don't manipulate DM1, because there were always 2 neurons which come from DM1, they survive here. But when they are there, you can see here in the second and third panel, they mistarget to the different layers, to the ventral layers. So that means E93 ECR signaling during development is very important for the formation and also for the identity of these neurons. So then, when you have 2 kids, you lose sleep, right? This is the second one here. So what do? So I'm a developmental biologist. I don't know anything about behavior. I wanted to study easy behavior. And for me, I thought sleep is easy, but it's not. It's very complex. So Krishna did make these 3D printed tools, but I was not confident as a developmental biologist because we need like very defined answer. So what I did is I reached out to the sleep doctors, and on right, here's Matt Kaiser, who's a neuropsychiatrist, studies patients, but also studies sleep in flies. And then Jenny, who is working as a scientist in his lab, and Buddha, who is now a faculty at Bucknell. Okay, so how do you track fly sleep? Again, now I may struggle here because this is behavior and I'm a developmental biologist. Usually there's a tube and there is infrared light. If the fly stays on one side, doesn't cross the beam for 5 minutes or more, it's considered as sleep. It's the activity monitor. And the Nobel Prize for sleep was given to Michael Rosbash and other people on this essay. And the flies sleep 2 times. One is siesta. This looks like it's evolutionary conserved. You can see here in daytime sleep. And also the longest one, which is deep sleep, happens during night. So there is day sleep and night sleep. So since they were sleep-promoting neurons, we thought maybe sleep behavior will be impacted. So what was a surprise to us is if you take this 5-minute threshold, there was no difference between 2 controls and pink, which is, or red here, which is knockdown. Neither in day, night. The bout number on the bottom left and the bout length also was not different from each other. So we were kind of surprised, why is there no difference between control and when there is no sleep-promoting neuron? Then there were lots of, I don't know how many of you follow central complex sleep studies. There were lots of studies where they show there are other neurons which are in the ventral nerve cord, which are important for that sleep, but we show that they are still there. But then there were these 2 papers where they show that there are different stages of sleep in flies. Like in humans, we have shallow sleep and we have deep sleep. So in deep sleep, the whole brain gets less active. And then Buddha, who is also in the paper, showed that there are deep sleep stages. And more than 60 minutes of threshold of staying on one side is deep sleep. And you can see here there's a significant difference between, if you see left bottom, between control and RNAi. Although they sleep same amount of time, but the sleep is more fragmented. They have more bout numbers and they have shorter bouts compared to control. That means this E93 is important for sleep consolidation in flies. If you lose it during development, neurons are not there, the sleep seems to be more fragmented. So this is kind of take-home message from this part. During development, stem cells make later on these DFBs which end up regulating sleep behavior. But I didn't tell that what does the same stem cell do early on. This is collaboration with Kathy, who is on left here. Kathy Nagel, amazing neuroscientist, 2 undergrads, Alexandra, and my second PhD student, Aisha. She graduated and is now a postdoc at Genelia. So kind of this is a short version of this paper. You can read more here. We show that same stem cell, which makes DFB, sleep-promoting neuron, also makes VFB, which goes to the ventral layer early on. VFB means ventral fasciate body because it goes to the ventral layer. Same stem cell. And then we show that early factor, which expresses in young stem cells called IMP. You lose IMP here in the second panel. Red is tachykinin, which is a neuropeptide. Green are VFBs. If you lose IMP during development, you lose these VFB neurons. If you misexpress IMP on the right side here, you get more of these neurons. That means IMP is necessary and sufficient for the formation of these neurons. And you can see, you can also assay the olfactory navigation behavior. These are 3 different flies. When the odor is not on, you can see gray. When odor is on, they will go towards the odor, which comes from top. They go against the wind, which is purple here. And when the odor is off, they wander, which is green here. So compared to control, you can see on the right side, the flies, they don't go towards odor. Their olfactory navigation is completely abolished. You can see also in this head distribution diagram on the right. So that means early factor IMP specifies olfactory navigation behavior. Late factor in the same stem cell specifies the sleep-promoting neuron, which regulates sleep behavior. Anyway, this is kind of take-home message from the science part. Same stem cell over time. I hope I was able to justify that they make different neurons, which go different circuits, end up regulating different behaviors through differential temporal program. Okay, so I was supposed to also talk about my other ambitions, which I have been doing a lot. So how do I train next generation? I started JK Scientist when I was a PhD student in Germany to train the next generation from Kashmir. I started it, but it has been run by most of the amazing volunteers. So they do most of the work. It has mentoring program, outreach program. And recently we did one. I go Kashmir for a week and then 2 days are there. You can also, Raj, if he's here, Raj was also there this spring. Again, in New Mexico, I started Pueblo Brain Science Program in 2020, which is a big community program now. And we organize lots of stuff. And the focus is here, the New Mexico, and specifically we have collaboration and partnership with the Navajo and Pueblos. I also like to teach and train next generation because I cannot take more than 5 undergrads in my life, which is always there, 5. I started NeuroCure to get more people, more undergrads into science, and majority of them, 70%, end up in grad programs and research. I started Meet a Scientist to get opportunity for New Mexico students to meet and interact with diverse scientists, and many of them developmental biologists, fly biologists. Lee was there last spring, and you can be here on the right. And Lynn is also there, you see. And in these meetings, we have lunch, pizza, and we discuss life. And also, I organized Developmental Behavior Symposium, again, thanks to amazing developmental biology people, Chris, Jim, they all came there on free. 2024, I said, I have space, I have food, but I don't have money. They all came, developmental behaviors in New Mexico. In the next year, last year, we made bigger version of it at Genelia. So it's called the Development of Behaviors, kind of developmental biology for it, and meeting their behavior people. Okay, so this is again Matt, which I met at Chris's lab. In Chris's lab, he was a PhD, now he's a faculty at Bucknell. We stayed friends. He always comes every spring. So we organized 3 days workshop for high school kids, undergrads, and teachers in New Mexico. These are 4 different posters from 4 last years. And lots of amazing instructors who come from different places. And postdocs also, faculty send postdocs. There are some pictures here from the events we do. Again, we are part of the community. The James and Zia Pablo, they invite me to us to their youth festival. We go there. For example, here on the top, there is a governor, I cannot show, close to me, of James Pablo. And this year we did workshop, not at UNM, because the challenge is for them even to come from the reservations to our university. So this year we decided on the van to go to these places. So we did one day Samoa College, which is a community college in Farmington. Then we drove to Dine College in Tuba City. And then last day we did in James Pablo, which is 40-minute drive from UNM. So it has been very impactful. So this is a very powerful quote, which when we do surveys, so the youth of Navajo are the people who participate in this. I think this is what keeps me going. So very powerful from one of the students in our workshop last year. So I will leave it here. This is a collective message from all the people I work with from Kashmir to New Mexico. So they have this free board they can write. You can see here, they tried to bury us. They didn't see we were seeds. And this is a message which I'm giving you from New Mexico, from Kashmir, from all the oppressed people of this world. And I am thankful for all my collaborators. Kathy, I talked about her work. Matt, I didn't talk about Vivek. We are also now having amazing project where we see developmental activity, how the activity of the navigation starts during development, which may be in next SDB. And 2 graduated, Adil and Aisha here in yellow. Again, they are from Kashmir, so it's again power of mentoring. They come from JK scientists, now postdocs in future. Lots of undergrads and lots of mentors. I couldn't fit all. You can see here, lots of developmental biology people here. Chris Zoe, Christian Clem, Claude, Shashi, they are from India. Ron Whale, Kathy is behavior, and lots of, Alex Kolodkin is developmental biologist, Alex Keen, Vivek, Carla, all these amazing people, and all the FLA community, Pablos of Zia, James, who have shown trust in me and invite me to their festivals, and funding from NSF first, and private foundation, Solomon, McKnight, Brain Behavior, DOD, and recently NIH. Hopefully it stays. I feel very, anyway. So thanks to UNM and FLY. And these are amazing people in my lab. What else? I think the other message, which is very positive, is this. This is the other kid, the second one. It says discovery never ends from Woods Hole, right? The good thing about FLY is you can send them to the daycare, and this is the activity they had to do. And they gave the answer. Thank you very much.